So it's time-based, so most people try to let it only go 1 or 2 synapses. And it's very toxic. So I know someone that I used to work with in Catherine Dulac's lab, Steve Santora, is trying to develop one based on WGA. So I think we'll probably see some advancements in that. The other thing that people do to get one synapse anterograde is just to do rabies from the postsynaptic side, essentially. So it doesn't let you identify unknown targets, but it's sort of a hack, I guess, to get monosynaptic.
So there is one comment I want to make. So there is a new thing now that it's actually not that new, but you can do AAVs that do anterograde transsynaptic for 1 synapse. But what happens, so that's been known in the gene therapy literature for a long time, but what happens, it happens with extremely low efficiency. So if you do with something like GFP or channel rhodopsin, you see absolutely nothing. But if you do with CRE, then you do get this transsynaptic anterograde transmission. And it's not, and the mechanism is believed to be that AAV just sticks to the cell surface and just diffuses down and then comes off. That it never actually infects it never actually transduces the presynaptic cell. It just rides along it and then pops off and then transduces the postsynaptic cell. So there was a paper from this guy, Brian Zing, I think it was in Neuron, I don't know, a few years ago, that talks about it, Z-I-N-G-G, and I think that's his name. And the other thing I'll say is that if you do it with CRE, you'll get toxicity at the presynaptic injection sites. So because CRE is toxic, so we've in high doses. So we've done it with FLIP and we haven't seen the same toxicity. So it does work for just with low efficiency.
Okay, thank you.
All right, should we get started? Oh, I was gonna add, Luke, did you post the prompt for the final?
Post the what?
Oh, I just wrote like a Word document for like what the students should do for the final project.
I'm sorry, I don't remember that.
Yeah, so we'll get it posted, but basically, as we discussed, you just work in pairs, 15-minute presentation regarding a problem that you want to solve using the methods, and talk about the pros and cons of the methods that you're using, and you know, talk about how you would overcome any disadvantages. Okay. So we'll get that posted. You guys can self-pair for the final. And we'll send out a private Zoom link for that one for next Tuesday, May 5th is the date. Okay, so I'll get started for today. I have a fair amount to cover, but I'm going to try to focus on like foundational principles that a lot of the other techniques build on. And as you guys can see the screen, right? So as Luke and I were discussing, these manuals are referred to as the Molecular Biology Bible. So a lot of these techniques really took off and were you know, standardized for bench use in the early 80s, and these protocols were developed, and they have a lot of information about troubleshooting and how to set up. So a lot of it's dedicated to cloning, some to in situ, and some other like northern blots and things like that. So some of the early techniques that a lot of the next gen techniques still build on, some of the same principles. So today we're going to cover CRISPR-Cas9. As I mentioned before, we sort of briefly touched on inducible systems, but I'm going to talk about them in more detail with particular attention to tagging active cell populations, old style sequencing, PCR up through RNA-seq and single cell seq. And then if we have time at the end, we'll talk about in situ hybridization and MERFISH. And I just thought this cartoon was funny because, of course, all of the new information, this is actually the CRISPR constructs with the guide RNA and the CRISPR-Cas9 attached to DNA. And of course, everyone is saying, oh, these are molecular scissors, but in this case, it is not scissors. It's a blindfolded person with a bat. So we'll talk about some of the limitations and advantages of CRISPR. So of course, we already sort of introduced CRISPR. CRISPR stands for clustered regularly interspaced short palindromic repeats. And these were actually sort of bookmark pieces of DNA in the genome of bacteria. And within these areas, they would actually, the viral DNA or any foreign DNA that came into the cell would be put into these repeat sections. And then that way the cell would know that those were foreign DNA, and they would basically try to destroy them. So that was the significance of those pieces of DNA. And then the protein that would recognize the foreign DNA that was in between these repeats was known as Cas9, CRISPR-associated protein 9. And we'll talk about what exactly, there are other Cas proteins, but 9 is the one that got developed. It's site-specific. It can be multiplexed based on how they've developed the technology. And we'll talk about some of the improvements on CRISPR technology. So CRISPR sequences have been identified and studied since the mid-80s, and it wasn't fully recognized that they were part of this immune system, like what the significance of the repeats and the intervening DNA. It wasn't really recognized until 2005 to 2010. And then the technology really started taking off as a site-specific endonuclease for recombinant DNA purposes in 2012. So at that point, Jennifer Doudna and E. Charpentier proposed an engineered system based on the Cas9 system. So Cas9 was in a particular strain of bacteria, Streptococcus pyogenes, and it was a system that was based on a CRISPR RNA and a transactivating RNA that together would guide the Cas9 to the DNA sequence and allow the endonuclease activity. And so basically what they proposed was that instead of having these two pieces of DNA, that you could actually use them together in one piece of DNA to create the hairpin and then the homologous region. So that was sort of the initial advancement. And basically there are two, I think it's only two types of CRISPR systems. And in one case, I think that the RNA is like specialized and it's part of the protein complex. But in this case, this is a type 2. It was actually a separate RNA and protein interaction. And so that actually allowed for sort of making combinations as opposed to trying to transcribe everything together. So it's kind of a modular system that allows you to sort of put in your recognition system and then the same protein. So it led to a lot of advancements. And so just to get more into what the components of the system are, and this is specifically referring to the engineered system, not the innate immune system. But it's composed of, again, the Cas9 endonuclease, and it's targeted to a specific gene location by a single guide RNA. So that, again, that's the combination of those two components that are in the native system. The Cas9 protein are separately encoded, so that means you can sort of do different tricks based on what we've talked about already. One could be expressed from a mouse genomic locus, one could be infected using an AAV, or you could do both via a virus. So it's sort of, as I mentioned before, modular system that you can kind of mix and match, and this is also what allows it to be multiplexed. So one important component that's present in not only CRISPR, but some other site-specific endonuclease systems related to CRISPR are a PAM sequence, protospacer adjacent sequence. In this case, it's for original like CRISPR-Cas9 engineered sequences, it's 3 base pairs. The length and the position of the PAM sequence is a little bit different, and it can lead to different specificity and off-target effects for some of the other mutated forms of Cas9 or the ortholog paralogs of the system. So In the case of the 3 base pair PAM, those occur about every 8 to 12 base pairs. So in most cases, whatever guide RNA you're trying to choose against a matching sequence region, you're probably going to have it close to a PAM sequence, but you need to know where they are in the genome so that you can target a guide RNA appropriately. So the guide RNA must have the PAM sequence. In this case, it's on the 3' end of your sequence. The guide RNAs are usually fairly short, 20 nucleotides, and you need to check that they're unique to prevent off-target binding. And then, as I started to talk about in the last lecture, basically there's these 2 enzymatic regions that will cause a double-stranded DNA break in this particular CRISPR-Cas9 system. And so when this occurs, about 95% of the time, you're going to get non-homologous end joining. And this process leads to insertion deletion mutations, or known as indels. And indels shift the reading frame, and that leads to a mutated copy that usually is not functional. In the case of homologous recombination or homology-directed recombination, known as HDR, if there is a homologous sequence, a lot of times it tries to use the native DNA, but if there's a homology sequence that's provided as a donor, it can use that. So in the case of a double-stranded break, it's only going to happen about 2 to 5% of the time. It's usually a mechanism that's used when there's a 3' overhang. So with CRISPR-Cas9, I thought I had this citation here, but I might cite it in the next slide. Basically, even in the cases where you did get homology-directed recombination, which again was a small percentage of the time, basically the CRISPR-Cas9 kept working and it kept making new double-stranded breaks even after the repair was made. And so there were insertions, deletions in the HDR portion, or the donor DNA portion that you were trying to introduce. So I can talk more about how they overcame that. But just to get back to some of the different systems of Cas9, there are some other orthologs that have been identified from other species of bacteria, or we'll also talk about the engineered ones. So the original Streptococcus pyogenes is notated with this SP. So if you see in, for example, the AdGene catalog, if you see SP-Cas9, it's talking about this particular Cas9. And the PAM sequence going from 5 to 3' is something followed by a GG. So this particular enzyme takes 4.1 kb. And as we talked about in terms of trying to express something from AAV, that's basically your whole coding capacity. So one advancement that was made that made viral delivery of Cas9 easier was the identification of an ortholog from Staphylococcus aureus, and this is noted as SA-Cas9. It has a different PAM sequence, it's a different length, so there's going to be some different places that you can actually use a guide RNA. So when you're building your guide RNA, you need to pay attention to which Cas9 system you've chosen. This one is shorter, and so it leaves the possibility of expressing both your SA-Cas9 enzyme along with a guide RNA, because again, the guide RNAs are fairly short. And these SA-Cas9 was described in 2015, and they've already got some constructs available on AdGene where they have the SA-Cas9 as well as a backbone area where you can just go in and clone your guide RNA. And I think you can either do, I believe it's like 1 or 2 guide RNAs along with SA-Cas9, or if you choose to deliver the Cas9 separately, I think you can put up to 4 guide RNAs into a single AAV vector. So there have been some other improvements on the Cas9 enzyme to allow enhancement of targeting, and essentially that means to enhance the specificity or efficiency of the enzymatic action. And so one thing they've done is that they've made it so that there's only one enzymatic sequence. So that's the nickase. So instead of making a double-stranded break, it makes a single-stranded break. So since you only have one break, you actually need a pair of the Cas9. So you'll get a break on each side. And as I mentioned before, this is going to lead to that overhang that's going to prefer HDR. So that's going to be a better system in terms of introducing donor DNA. And then another one that I think is actually very interesting in terms of not just modifying DNA, but doing other types of manipulations using the fact that Cas9 is site-specific and it goes and binds in a particular place on DNA is the dCas9 focal. And dCas9 just means that it's dead. It doesn't have any enzymatic capability, but in this case, they've conjugated it to this focal domain such that when there are two of them, now they make a double-stranded break. And so basically you can imagine other applications of the dCas9 where you could actually just have something bound. And so that leads me to the next one, which is using this mutated Cas9 to actually act, instead of using it for its enzymatic properties, now tethering it to a scaffold. So there's one known as SunTag, which basically just adds a bunch of these activators, and then that could potentially lead to transcriptional activation at your preferred site. And there are some other methods to use this same mechanism of it binding in a particular place and then recruiting specific elements. And they've also now tethered it to a repressor, and that way you can have sort of site-specific repression of gene expression. And I think there's ways to now introduce like epigenetic modifications site-specifically. So as we touched on last time, one thing that you will be using site-specific modifications using homologous recombination would be to make mutant mice. So for CRISPR-Cas9, this is quote unquote one-step mutant formation. So basically here, you can use Cas9 to directly edit the genome of the embryonic stem cells. So you don't have to go in and look up these sequences, make a cloning cassette or vector, and then go put it in. You're actually making a mutation at that point at the native DNA. So you're not swapping out any DNA. And so in this paper in 2013, they actually showed making, I think it's a triple knockout of these TET enzymes. TET enzymes are part of the enzymatic pathway for DNA methylation. And actually, just as a side note, that's a project of a PI at Albert Einstein, Milad Dewali, and he was one of the authors on this paper. So basically, they were able to just make all of these mutations in one step and get a triple knockout in 6 months. So it's basically a very easy way to get site-specific editing, and it's directly in the ES cells. And I forgot to also put the first report was even multiplexed gene targeting. So basically, most of our discussions are regarding how to use these techniques in the brain. So these are just a summary of some of the different ways that you can get components of the Cas9 system expressed in the brain. So of course you can take advantage of the large packaging capacity of lentiviruses and use traditional Cas9 with gRNA. So lentiviruses are often used for cultured neurons. And then as I mentioned for AAV, you can use the smaller SA Cas9 along with your guide RNAs. And then, of course, this is not for in vivo, the next two, but these are just some of the ways that people are getting the components into the brain. And then in my lab, we have this mouse. It's a conditional Cas9 system. So basically you have a lock, stop, locks, and then you have your SP Cas9 along with a cleaving protease, and then the EGFP. So you get tagged neurons where the recombination has happened, and all you have to do is cross it with a CRE line. So then once you have your CRE mouse crossed to the Cas9, you're going to have Cas9 expression in those cells. That by itself doesn't do anything. So then you have to go in with a virus and introduce your RNAs, your guide RNAs. And so that brings me finally to wrap up the section on Cas9 is to how to design your guide RNAs. So basically what you want to do is enhance the efficiency. So actually do what it's supposed to do. So not put a guide RNA and then not have something happen. So either the knockout, the indel mutation or the homologous recombination. And then of course you want to go for specificity. So don't do something that you didn't ask it to do. So these would be off-target effects. So here's I like timelines of things. It helps me understand stuff better. So there was a good recent review showing some of the tools. So basically, I think the first and foremost step that people use in determining what their guide RNA should be is just computational. They just go in and look up against whatever their host, the target organism, check homology for a short guide RNA to see if it's going to be specific, if it's going to bind somewhere else. So you can play with the length of it. You can play with which PAM sequence within the gene that you're going after. So there's tons, more. There's all these different computational tools. And then the second step would be to actually go into cells, test your construct before you put it into a live animal. For the most part, I think people a lot of times skip that step and just cross their fingers that whatever they checked the predicted binding to be will be right. But probably it will save time if you actually go in and validate using cell system before you put it in the brain. So next we'll talk about inducible systems. So first I'll just remind, so we kind of touched on these in the last lecture. The Gal4 UAS system is mostly used in flies. So we're probably not going to talk about that, but some of the principles are really similar with some of the inducible systems that are used in mammals. So I just wanted to mention it, just, you know, if you're sitting at a talk and you're thinking, what is this? So basically Gal4 just promoter specific inducible system. So you put a different promoter upstream of the Gal4 and then it drives, in this case, T is for some kind of transgene. So similarly, the tetracycline transactivator and tetracycline response element are sort of a similar system. In this case, it's in the presence of doxycycline, your transgene would be off, and in the absence, it would be on. There's also a reverse system where it's T on with dox and T off without dox. And there's some reasons why you might choose one over the other. Yeah, here shown here. And then, of course, as we talked about, you can have a promoter driving CRE, and then you can have a flex or floxed component downstream, and then you can have expression of your transgene with CRE, or you can have something happen, site-specific changes, either deletion, activation of something, reporters, all of those things that we talked about in the last lecture. And so in terms of what we're talking about for now is combining TET expression with and having the specific thing that it's driving be CRE. So then you can combine it with other things. So you can have direct tetracycline, or another system that we'll talk about is the tamoxifen bound CRE-ER, CRE-ER-T2. So these systems, instead of having them directly drive some kind of reporter or something, you have them drive CRE and then have CRE have an additional layer of control over something. So in this case, it would be TET on. So in the presence of dox, it's going to drive CRE. And then here with the mutated CRE, CRE-ER, the mutated estrogen receptor, which can't localize to the nucleus except in the presence of tamoxifen, then it will go and it will drive expression of something. So as we mentioned before, there's CRE-on and CRE-off, and we'll talk about tamoxifen-inducible CRE. So of course, no tamoxifen, you don't get expression of CRE. And then here, with the presence of tamoxifen, CRE-R can go into the nucleus. And in this case, it's removing a stop codon and having expression of a reporter. And then one thing that I do want to point out about this tamoxifen-inducible CRE, and it's something that goes back to what we discussed in the last lecture, is you can take tamoxifen away. It will talk about some of the dynamics of being able to label with tamoxifen. But what you've done in this particular case is permanent. So even in the absence of tamoxifen, those cells are still going to be green because what you did was permanent. So just keep that in mind. So what are the complications that come from this? So by the way, I started using the new version of PowerPoint and it suggested some like formatting for me. So if you notice some of my slides look different, I went with their suggestions, and in some cases I thought they were pretty nice. I've been in quarantine a long time, so some of these things really entertain me. But some of the complications that might arise are leaky systems. So this means that in the absence of the chemical activator that you're using, you're still having expression. So there are ways to maybe increase the signal to noise, but basically there's always going to be some concern for leak in the system. And then the other thing you have to consider is the drug half-life. So most of the time in the case of tamoxifen, it's being injected, and we'll talk about some of the systems next, the specific systems. In the case of DOX, it's usually given in drinking water or food. So how it accumulates in the cells that you need to do recombination in is going to have a ramping up, and then it's also going to have a ramping off. So the time course of actually controlling when your manipulation happens, obviously it's more than a constitutive system where you have it when, you know, it happens whenever that gene developmentally comes on. So you have more time control in that sense, but it's also on the order of like hours or days, depending if you're injecting or giving in the food. And then finally, you need to think about the effects of the chemical on your actual behavior or on the cells. So obviously certain doses of these chemicals can be toxic, but in the case of tamoxifen, it's obviously acting on estrogen receptors. And so if you have something like a sex-specific behavior, which I study a lot of in my lab, you might be concerned about giving basically something that binds at the estrogen receptor. So you need to have controls in place for administration of the chemicals. So there was a good review a couple of years ago on some of the mechanisms for tagging active cell populations, and this came from Leach and Lowe's lab. So they are not biased to mice. They also have some systems that are used in flies. So they break down these systems into sort of how the promoters are, how the tagging is happening. So there are several that are based on immediate early gene expression. So FOS, ARC, those types of things. And so we'll go through some examples of those particular systems. And then there's a couple systems now where instead of basically endogenous IEG, they've actually made a synthetic IEG, so that's ESAR and RAM. And then there are some that are calcium-based, and there are some that are actually based both on calcium and light delivery. So that's, in this case, they had shown Campari, but there are a few others now in development that we'll talk about. And then this one is mostly used in flies. It depends on the UAS. So this is another one of the pushed designs for me. So basically I couldn't find any cool graphics and the slide was all text. So I put some canes over here to highlight the ridiculousness of all the acronyms that we have to discuss in the next few slides. So For immediate early gene-based systems, one of the earliest was the TET tag. Then came TRAP2, CANE, and we'll talk about these three. And then VGATE is very new, so I haven't had a chance to really review the literature on that, but I think there are even probably more coming out. So TET tag was introduced by Mark Mayfer's group. And I think this was the one that was made famous for introducing the idea of engrams and ensembles of ensembles of neurons in the hippocampus that are involved in fear memories. So different kinds of memories. So, and then I think it was further developed by Steve Ramirez and colleagues, and in terms of like engram research. So essentially, this construct has a FOS promoter that drives the tetracycline transactivator. Then TTA binds and allows transcription either directly of a reporter downstream of the TET operon. You could also put in a TTA cree and then have an additional layer of control. But in this case, they directly drove a reporter. And I think one of the weird things about this, I've used this actual mouse, and it also, while it drives TTA from the native promoter, it also drives GFP, which can be actually kind of annoying if you want GFP to be your label instead of LAC-Z if you have TETO. So you have to use TETO tomato essentially with this mouse. So basically, in this one, you keep the mice on doxycycline food or water until you want expression. So then you have to take the mice off the food or the doxycycline treated water. And one thing that I learned having used the mice is that the dox food is green. And so the way you tell if they have it in their system is the color of their poop. So that's a pro tip. What color their poop is tells you if they've got dox food in their system. As far as the water, I think if you put it in the drinking water, it's a little bit faster in absorption, but no cool poop to look at. So in the case of taking them off, I think it took about 2 days to get them off, and then a couple of days also to on the other side, to put them back on dox and get the promoter to stop. So it was a little bit tricky in terms of trying to get something expressed for, in our case, we're trying to use it for hypothalamus, but I think it has some utility, and of course it was sort of the propulsion of other people to start looking into how you could tag active cell populations. So the system that I've decided to use in my lab is coming from the second generation of the TRAP system. So TRAP for transient, oh man, am I gonna remember what, I thought I had TRAP. Oh yeah, targeted recombination in active populations, sorry. TRAP stands for something else later in the talk, so it's pretty cool that there's the same acronym for two different things. So in this case, they're taking advantage, instead of a TET operon here, they're using the CRE-ER-T2. And so of course, without tamoxifen, nothing is happening. With tamoxifen and activity together, because this is downstream, in this paper, they developed both ARC and FOS CRE-ER. So that will activate transcription of CRE-ER-T2, but of course CRE-ER-T2 can't do anything unless it also has tamoxifen. And so in this case, they used a reporter, which was the lock-stop-locks tomato. And so basically for their proof of principle, they basically did some barrel cortex experiments with whiskers. So in intact, you can see with tamoxifen, you get all of these whiskers labeled. If you take one whisker away, you lose immediate early gene reporter expression, specifically in that barrel. And then if you have all but C2 plucked, now you only have activity there. I think in the initial case, they had made it using a transgene, but I don't recall anymore. But with the second generation TRAP2, it was a knock-in, and so now there's into the ROSA 26 locus, and now there's no disruption of endogenous CFOS expression, because that was a problem with the original that was reported in 2013. And then the other thing I wanted to mention is that tamoxifen is not actually what is functional. It's actually the hydroxytamoxifen. So when you inject tamoxifen, you're waiting for that conversion to happen. So if you directly inject with hydroxytamoxifen instead of tamoxifen, you can control the dynamics of the expression, so of this allowing your Cre-ERT2 to now go into the nucleus. So here you can see the time window is much tighter. You only have to worry about maybe 6 hours instead of up to 24 hours after your first injection of the drug. So that's pro tip for TRAP. Cane. The reason that I included cane, I think it's a little bit of a pain in the ass to use. Oh, sorry, I was gonna not curse when I lectured, so I apologize. Someone put a bleep on the recording. So it can be kind of a pain to use because it requires activation of your immediate early gene followed rapidly by the injection of a virus. However, I think that there are some advantages to the system. So I think it might be a little less, I think you have much less concern for leakiness in the system because you are going in with a virus. And then the other reason that I wanted to mention this is because instead of being inducible in the sense of the Cre-ER or the tamoxifen, in this case, you're getting active populations tagged using the mechanism that we discussed in terms of monosynaptic labeling. So in this case, they're using FOS to drive the TVA receptor, and then they're basically pseudotyping a lentivirus with the they're pseudotyping it such that it will bind to the TVA. And then that will drive your gene. So basically, yeah, sorry, that's pseudotyped with the VSVG. So that will go specifically into the neurons that have now expressed TVA. So once you get expression of the TVA, you can go in with the virus and then it will permanently recombine. In this case, they were using it to see what neurons in the VMH were important for aggression behavior. So if they do, I forget what SF stands for, but this is social fear and aggression. So essentially what they found, again, you have to go in quickly with the virus after your behavior to infect. And what they find is that if they look for CFOS and they look for where they got recombination, it's only about 60% of the cells. And I'm not recalling off the top of my head what the efficiency is for some of the other inducible systems, but it's not so great. So if they do the same behavior twice, they capture those cells using cane, or they look at CFOS, they're only getting about 60% of the cells. Same goes if they do AG and AG. And then if they do different behaviors, basically they were trying to show in this paper in 2016 that there were different cell populations in the VMH that were responsible for social fear and aggression, but they do have some overlap between those populations. So this was another suggested format, and I thought it was great with the ram in the truck. So I think you know, the company should probably contact me for this proprietary design. But essentially, they're synthetic activity promoter systems. I'm not going to talk about eSAR. Supposedly, RAM is better. I don't know a lot of people using RAM, but we'll specifically talk about RAM. But what does eSAR stand for? It's an enhanced synaptic activity responsive element. And then RAM is much less informative acronym. It's a robust activity marking, but we'll talk about what they did. So in this case, they made a bunch of different promoters, and in the initial rounds, they chose a bunch of different ones besides FOS. I think they tried some C-June elements. I think they tried ARC, but essentially they settled on FOS, and then they said that between all of the immediate early genes, a lot of them had sort of a consensus enhancer module, so then they just sort of stuck it in a bunch of times and in different combinations for the ones they tried. And then when they tried to look at induction of the gene after a reporter after an activity, the one that was named RAM was the most efficient at getting activity. And then they compared it to eSAR after, driving the cells like crazy. So they get a bunch of, in this case, they're using luciferase as the readout. Compared to eSAR, so supposedly this is an improvement. So then basically they used this to drive tetracycline transactivator, and then now they can put it into one of these inducible systems. So basically you can see when they tried it, I think in H, this is cell culture, but basically when they drive a lot of activity, you can see the reporter. So I think, maybe I have a slide on it, but I think a lot of these viral components that were reported in this paper in 2016, I think they're available on AdGene now, if you're interested to use it. So then some of the calcium and hybrid systems where it's dependent on calcium and photoactivation. Campari, which is calcium modulated photoactivatable ratiometric integrator. Have you guys already talked about that in the other lectures? I think you touched on some of it. And then there's another one known as CalLight, Flare, and then FOTSeq, not to be confused with the drink Campari. So in this case, they showed in 2015 that they basically could have this sensor that essentially in the presence of high calcium would switch. So basically here you could go back and cells that were activated, now you could get to change from green to red. So in this case, they were using a visual task, and they could show that the cells that were activated, now they could get basically this reaction to take place with both calcium and light. And this requires a 2-photon setup. In the case of CalLight, instead of being that kind of reaction, basically now they've tethered tobacco etch virus protease. In one case, it can't bind when there's no calcium. In the presence of calcium, now the C-terminal side and the N-terminal side of TEV can come together, and it can cleave and release TTA to allow it to bind to TEDO. So you can see that the reporter is not being, in this case, it's GFP. It's not being driven only with activity. It can't be driven only with light, but it's the combination that starts driving the activity. And then if anybody was at my recent journal club, you already heard me bloviate about FOTSeq for like way too long, but this is physiological optical tagging. And in this case, they were using it for downstream applications of qPCR and RNA-seq, but essentially it's still a tagging method. So they have their 2-photon system. Basically, they can see what cells are active using GCamp, and it's tethered to a photoactivatable M-cherry by a 2A linker. So in the same cells that were active, you can basically find them and then go back and put create a mask of where you saw that activity and specifically photoactivate. And that allows expression of red reporter in the cells that you saw as active. And then you can put that for downstream applications, which we'll talk about next, RNA-seq or qPCR. Yes, so here you can see all of the available resources, either the AdGene constructs, the Jackson mice that are using all of these. So this was a pretty good recent review summarizing some of the tools. And I didn't want to spend too much time, since we're sort of oriented towards, populations of neurons based on their activity. So I didn't talk about the synaptic stuff, but there are some tools where you can label synapses. So with that, we'll take an intermission, but I wanted to give a plug for Dr. Juleson's review on some of the considerations you need to use when you're trying to use some of these cell-specific targeting methods for neuroscience. So go and read that paper during a 5-minute break, and we'll come back at 11:25. We'll get back to it. We have quite a lot to get through, but if we don't get through I don't want to go like so fast and confuse everyone, but whatever we don't get through this time, we can pick up for next time for our last official lecture session. Okay, so in terms of sequencing, again, I like to have a little bit of historical perspective to give some kind of context. But basically, as we probably all know, the DNA double helix was described in 1953. And then the first recombinant DNA was shown in 1972. Sequencing methods, Sanger sequencing, that we're going to talk about in the next slide, started around 1977. And that allowed for complete sequencing of viruses and phage. We'll discuss PCR, which came about in 1985. And then in 1988, of course, we talked a little bit last time about the parallel efforts in industry and the government for the Human Genome Project. 96, complete sequencing of S. cerevisiae, the first eukaryotic genome. C. elegans in 1998. Drosophila in 2000. We talked last time about how the first draft sequence of the human genome was in 2001. And of course, then we talked also about the other, whatever 100 genomes project it is, and that started to produce mouse, rat, dog, chicken, chimpanzee, rhesus from 2002 to 2007. And of course, now we're in the era of personal genomes. And then the other thing that is going on now is not only having sequences for these animals, but also trying to identify the SNPs. So not just one mouse, but hundreds of mice, whole exome, whole genome, de novo mutations, especially related to human disease. And then looking at not just model organisms, but other systems. And so, of course, why did this explosion take place? We're going to talk about the technologies behind this massive increase, but essentially how much sequencing you could do just hugely shifted with the introduction of massively parallel sequencing platforms known as next-gen sequencing. So we'll talk a little bit about the pre-technologies and talk a little bit about the post-technologies. So original Sanger sequencing described in 1975. So essentially here what they did was they took a template and they started to transcribe it, and then they had a random mixture in separate reactions of some unlabeled NTPs and some that were labeled with phosphorus isotope P32. And so in some of them they had it, and some of them they didn't. And then they, for the specific each nucleotide. So of course there's 4. So then essentially having this tagged one would halt the progression. And so that progression would stop at random points for different templates. And you could basically run that out on a gel and see where you got a band, because it's giving off radioactivity. So it's going to create a signal. So that was the original sequencing. Today we don't use radioactivity. Today it's based on capillary electrophoresis, but the principle is still the same. You have these are dideoxynucleotides, and because they're dideoxy, they stop the transcription reaction at different points again, and then depending which nucleotide was at the end when it terminated, you get a different fluorescent signal. Gets run out on a teeny tiny gel in a capillary, and then that gets read out in sequence depending on the size. So that's still used today, the general principles behind Sanger sequencing. So next, and this is basically chronological. There's not really a reason why I'm talking about PCR next, because Sanger sequencing is more related to some of the next gen applications, but I'm going to talk about it because I think it gives some of the context. But basically it was invented by Kerry Mullis when he was working for Cetus Corporation. He conceived of it in 1983. As we were discussing, a very significant year in molecular biology. It's when the Bibles that I showed on the first page were being written by Tom Maniatis. But here is, he won the Nobel Prize 10 years later for this discovery, so that's a pretty quick turnaround. And it basically allowed you to amplify DNA in vitro over and over again based on some tweaks that they made to the chemistry. And so you guys know I like to do a deep literature dive. So basically I found this guy with this amazing mustache, and I'm pretty sure my dad has this exact same watch from 1983. So you don't have to have a mustache to run PCR, only in 1983. But basically there's a famous story about the origin of PCR. And I never knew it was actually a Scientific American article. I had just, heard this as a legend that, Kerry Mullis got the idea as he was driving down the highway in California. And I think I didn't have a chance to read, this is a 10-page article, and I found the PDF online, so I'm definitely going to give it to Luke to post for you guys, because it's just amazing to read. And of course, in addition to this photo, there are some amazing illustrations from the 90s that are just so fun to look at. But I think I remember there being some story about waves and surf, but I didn't come across it. Apparently he was just driving down the highway with his girlfriend, and he said that winning the Nobel Prize didn't make up for the fact that once he got the idea for PCR, he was in the lab and his girlfriend dumped him, and it just wasn't worth getting a Nobel Prize for. Anyway, why am I going on and on? It's because Kerry Mullis grew up in South Carolina, the same place I grew up, and I was born in 1983. So, basically I think my birth inspired him because it was April, and, it's just the stars aligned. And so I feel a very personal connection to PCR. And just FYI, he grew up in the Midlands, so, not far from where I actually was born, because I was born in Richland and that's where he went to school. So it's fate that I'm here today talking to you about PCR. So key ingredient, of course, that actually allowed PCR to be developed is Taq polymerase. What is Taq polymerase? Anything that has DNA is, well, with the exception of maybe, yeah, pretty much I think anything. Some viruses only have RNA polymerase, but everything that has genes has DNA polymerase. It was isolated from Thermus aquaticus, and that's where Taq terminology comes for, and it's very thermostable. So the range, it doesn't denature even at 95 degrees. So that's a pretty critical component of being able to do the PCR reaction. And so it was isolated in 1969 from Yellowstone National Park hot springs. So this is the actual molecule, and these are some of the hot springs where it lives. And it was designated Molecule of the Year in 1989, which was not a prize I was aware of until I did the research for this. And then of course, there are some really core components of PCR. So I apologize in advance if I'm boring anybody, that you've already heard a million lectures about PCR, but essentially the components are the Taq polymerase. The polymerase will bind in the presence of double-stranded DNA. So you melt the DNA so it falls apart, and then you put on these particular probes for specific sites. Where the probe binds, it becomes double-stranded so the Taq can bind. And then it starts adding dNTPs. And then you have a buffer that has a specific magnesium content and pH, and all of that allows for the reaction to happen. So I'm sure we've all seen this boring diagram, but I'll just have it here to remind you to keep in mind as we talk about some of the other applications that are based on the core technology. So basically, again, as I said, you denature the DNA, let it fall apart, you anneal the primers, and then where those primers have bound, now Taq can go in. When Taq binds, it starts adding those nucleotides, and then you can start the process again. So from the original one, now you have the 2 original strands and the newly synthesized strands. Now you go back to this step, and 4 become 8, and it becomes exponential at some point. So there's a good text here in terms of what the reaction is. So again, denaturation happens between 94 and 98 degrees, typically 95. So the entire reaction starts with about 5 minutes of that once you have your DNA and all the important components with it. And it breaks the 2 strands, but again, it doesn't damage the Taq. Then you lower the temperature, and this is kind of annealing, is we're going to talk about it down here, but the annealing temperature really depends on the melting temperature of your primers and how your primer is designed. We'll talk about that. It's pretty short, 20 to 40 seconds. Elongation is then where your Taq is actually going along and adding those NTPs. And usually this is more dependent on the polymerase, and that occurs around 72 degrees. So this step, you can basically have low yield if this step isn't long enough. So you might want to play with the length of that step, and we'll talk about that a little bit. And then theoretically, the amount of your sequence will double each time. And that will lead you to having to have a certain number of sequences before you get enough product. Usually in the case of, day-to-day genotyping PCR, for example, 30 to 35 cycles is usually how long you need. So something to consider in terms of template design. So on the NCBI website, it will create primers for you based on your sequence of interest. So you just put in the gene you're looking for, and it'll give you different choices. It'll give you the 5' and the 3' side. But some things that you might want to think about are, how long they are and how long the intervening sequences between your 2 primers. So all of those things can be optimized based on your gene. The GC content, so GC is more likely to make secondary structures and bind to each other. So in that case, you may need to change your annealing temperature, or what you can do to overcome the secondary structures that are formed by the primer sticking on itself. You can also add DMSO, glycerol, or betaine, and that will lower your denaturation step and lead to less secondary structure formation. So just a couple basics in terms of PCR and how you might troubleshoot. So not long after that, quantitative PCR, also called real-time PCR, was developed. I think, again, Carrie Mullis was involved in this. It uses a polymerase that fluoresces when it's bound to double-stranded DNA. So basically, if there's not primer binding, the cybergreen does not bind, so it's not fluorescing, and then when it binds, you get fluorescence. So in this case, you're taking, instead of looking at the end product, which is what you would run out on a gel for regular PCR, you are shining a light beam before you do that denaturation step and start again. So for each one, you're taking a fluorescent measurement. So obviously, as the product starts to accumulate, you get this exponential rise in fluorescence. Then it starts to approach a linear phase, and then there's a plateau. So hopefully you have enough cycles. So typically in qPCR, you're going to need a few more cycles to make sure you've really saturated your experiment. So you want to make sure you have a plateau phase. In my experience, for most primers, 40 cycles is necessary. So then when you have different, and here I'm showing what a standard curve looks like. So typically you can do spectrometry on your sample, so you know how much DNA you are putting as input. So basically you can make dilutions of your DNA, and you can see essentially when it crosses this exponential threshold known as CQ, what cycle number does it cross that threshold of fluorescence. And so for each primer set, you're going to have a particular threshold line for the amount of fluorescence. And so when it crosses that line, obviously the earlier the cycle number is, it means the more DNA quantity you had. So then you can go plot your known, full dilution, you know what that quantity was, you know what the CQs were, and then you can basically back calculate the quantity that you had. This is going to be different for every primer set, and it's going to be different not only because of the specific primers that you choose, but also whatever the characteristic of the gene is. So a lot of times as controls, we use ribosomal RNA, and it's very, very abundant. So basically that's going to have a really low CQ value. So just does anybody have questions or comments on PCR? No, you guys are bored of PCR. Okay, so reverse transcription is a tool that you can use to basically measure RNA expression. So basically PCR can be used for both RNA and for DNA. What I've been describing is more like what you would use for genomic DNA. You can also start with a step of reverse transcription if you have RNA. And there are some things you want to keep in mind. So confusingly, this is also RT-PCR, which is different from RT-PCR real-time, but you can have quantitative reverse transcription or qRT-PCR. But anyway, just keep in mind what the particular acronyms are standing for so you can understand. So basically the first step would be to isolate and quantify your RNA. The essential step is your first strand synthesis. So from your RNA, now you're getting complementary DNA. So that's stable, and that can actually be used in the PCR. So once you have your cDNA, you can go in and remove all of the RNA and then basically amplify using the traditional PCR. This can be done back in the day when I learned it. It was like a couple of steps, but now I think there's like some sort of, you can put in a reverse transcriptase and your TAC at the same time, and it's basically one step. It's just a kit. It's not that hard. So basically you just want to start with high quality RNA and then, do your cookbook steps. So how are we getting the RNA? So in most cases, if you recall, again, we're starting with RNA, so it's different from starting with DNA. So most eukaryotic RNAs are going to have a polyadenylated tail. So one trick you can do to just get your first strand synthesized with your reverse transcriptase is to create that double strand with oligo DT. So that just means you have a bunch of T's. The T's will bind to the A's, and then you have the double stranded part. If you're working with a sample that's not eukaryotic, or potentially you're looking for non-mature RNAs, or for example, you're looking at viral, a mixture of like mouse and viral or bacterial, whatever, you might use a different kind of primer called random hexamers. So each is going to give you slightly different biases. So if you only use oligo-DT, you're only going to get mature RNAs that are polyadenylated. So that's one bias that you can introduce in your downstream applications, and first-strand synthesis is part of all of the next-gen technology that I'm going to talk about. So I'm going to try to point out where there are places. Some of them I'm probably not aware of, but this is one that can create bias in your downstream applications, what kind of primers you use for your first-strand synthesis. Okay, TACMan. I've never used TACMan. But it's basically another way to assess what's in your tube. TACMan can be a little more sensitive because you can multiplex in terms of the fluorescence and check if you have, in this case, you can see if you have a certain mutation, or if you're looking for SNPs, you can identify SNPs using TACMan probes. So essentially, in this case, it's not, your polymerase is not fluorescent, excuse me. And it's not the primers that are fluorescent, but there's this probe that binds on a region of interest within your target DNA, and it's bound on one side to a fluorophore, and it's close enough to a quencher that typically it's not going to be fluorescent. And then once your forward primer binds, it starts to transcribe. It's going to, so one of the things that, in addition to being a polymerase, there's nuclease activity of TAC. And so it will actually cleave off those probes that are sitting in the way of the polymerase activity. And then once it's cleaved away from the quencher, now it fluoresces. So you can basically have multiplex systems where you can see if there's, if you have one particular primer, you will get, you know, displacement of the quencher. But if you have, for example, a blocker that also contains a quencher that has a different SNP, you will not get fluorescence of your TACMan probe. So that's just one advancement that's been made, and you can use a qPCR workflow essentially to do your TACMan assays. So another technology that I personally haven't had the pleasure of using yet, but I think is going to get hot real soon if it hasn't already. I think one of the problems is just the availability of the machines, and of course there's a huge cost to getting all these new machines that you need to run things, but essentially there's something called digital PCR. Digital PCR doesn't have to be based on droplets, but I think there's a separate sort of digital droplet PCR, so you may hear both terms. And this was sort of described in the late 90s. I forget the people who were describing it, but basically you can take a tube that's full of either complementary DNA, genomic DNA, or RNA, and essentially you can separate it into like teeny tiny wells such that it's sparse enough that one or none of the templates is captured in these small, essentially, reaction wells. And then you do basically the same, you do PCR within each well. And so essentially in one case, you're gonna get tons of accumulation. So you only do one readout at the very end. So this is more like maybe regular PCR, but on a chip or something. So in some cases, you're gonna get tons of accumulation, and in some cases, you're gonna get nothing. So it's basically ones and zeros. That's why they're calling it digital. But it gets a little more complicated because you can actually multiplex it. Nonetheless, then you go in with your detector to see. And so essentially there are, you can use two different fluorophores in this case. And so there's a little bit that you can do in terms of like the actual reaction based on how the probes interact. Sometimes they'll be very high fluorescence, sometimes they'll be medium fluorescence, and you can have two different fluorophores. And so you can have basically 6 different readouts instead of just one. They come out as 1s and 0 for each channel, but you can have essentially 6 channels. In this case, they're calling it true quantitative. So in the case of qPCR, you actually have to go back to a standard that you've made. But in this case, it's truly quantitative because the amount of fluorescence is going to tell you exactly how many, sorry, it's not the amount of fluorescence within the wells, it's the number of spots that you count in your array. So I think this could be really useful for certain applications. I didn't want to completely overlook microarrays. I feel like microarrays have gotten lost in the whole next gen era, but I just wanted to mention it in the mix with talking about some of the other technologies that are based on hybridization, because essentially that is what microarray is. It's hybridization of targets at like a massive scale. And I couldn't actually find the story behind it, but I think Pat Brown was actually like, he just acquired a microchip machine and then he made it put DNA onto chips as opposed to putting, I don't know, what is it, copper wires? I don't know enough about computers, but I couldn't find the story again, but that's the story that I heard about how microarrays came about, and that's why it's called DNA chip, because it's basically dependent on the same exact technology that computer chips are designed on. So basically you take your, again, you've got a sample, you're going to purify whatever it is. If it's mRNA or if you want total RNA, use random hexamers to do it as well. You still need to get your first strand, your complementary DNA, and then basically you shear it all up and you label it. You can do up to 2 dyes at the same time. So in the case of, I think the case that they were giving an example of is like cancerous tissue versus normal tissue. So you have one in green and one in red. In the past, when I've used a microarray, I think I had a control sample and you know, my sample of interest in my microarray. And then essentially you perform hybridization. So basically each of these spots contains a bunch of different probes. Probes being basically primers. So they're primers against different parts of the sequence of interest, and all the primers for one sequence are on the spot. And there are different, you can actually do some like kind of fancy things with microarrays. You can basically make them small and only put like olfactory receptor primers on them. You can put primers against different exons within that spot, so you can sort of have a sense of how much transcription, a quantitative idea of like how much transcription for each exon there is. But basically all of the stuff for the same one is within a spot, and then you have all these different spots, and then you wash all of that labeled DNA over, and it's basically a hybridization step. If we have time, we'll talk about in situ hybridization, but basically this is a little bit like in situ hybridization. And then so you incubate your DNA at a high temperature on the DNA chip, allow it to bind, and then you just image and see how much red versus how much green you had on each spot, and that'll tell you how, it's basically semi-quantitative, how much binding you got against those primers. So DNA microarrays, the last time I used one, I think it was like $250, so they're pretty cheap. So if you don't need to look at too much, you know, sequence depth or something like that, you can skip RNA-seq and just do a microarray. Or for example, like, I think there's one that's just against neuropeptides. So that's something that's, you know, can be useful and low cost, and it's pretty simple to do. I don't know if people still have microarray machines, but it has its place. So now we're going to talk about next-gen sequencing. So next-gen sequencing basically is referring to all of those technologies that are massively parallel. So whereas Sanger sequencing, you have all these different products that need to be run out on a gel, so each capillary has to be read individually for one sequence. These are basically based on similar types of ideas, but now you're doing them in massive parallel. And part of what helps them be parallel is a system similar to what the DNA chip is. So it's kind of taking a lot of these different ideas and bringing them all together. So next-gen sequencing, most people think of sequencing by synthesis. That's sort of like the most, as far as I know, it's the most prevalent one. There's also sequencing by ligation, pyrosequencing, which I'm showing here, the workflow for basically pyrosequencing, and then ion semiconductor sequencing, which is similar to pyrosequencing. In pyrosequencing, you're having light readout, and ion semiconductor sequencing, you're getting readout of from like hydrogens or something. So basically, the idea is that all of them can do sequences all at the same time. In the case of pyrosequencing, you can actually get much longer sequences than some of the other ones. I'm gonna go most in depth for the sequencing by synthesis, but if you're interested, others of these methods or even Sanger sequencing are used to validate, because basically all of them have their technical limitations, either how long the reads can be, how error prone it is. So typically you do some sort of validation with another technology. Okay, so basically the work, oops, I didn't finish the title here. This is sequencing by synthesis. All right, and so basically the workflow, again, the sample prep is pretty similar to what we've talked about for qPCR and microarray. You still have to have your first strand synthesis. There are some people in the camp that you shouldn't amplify your DNA, but I think some of the technology can basically account for the fact that you're amplifying. And then there's something called library prep, which we'll go into more detail, but essentially you need to put adapters onto you shear up the DNA and then tag the RNA and tag it with some adapters. And then the first step, once you actually go onto the column, is cluster generation. Then you actually proceed with the sequencing step. So the cluster generation is actually much more key. We've already talked about the sequencing steps, how that can work. And then the big pain in the you-know-what is analysis. So you can get these massively parallel reads. You can spend 2 weeks getting all this data, and then you can spend 6 months analyzing it. So it's not that easy to explain. So I am just going to show a video for like 5 minutes. So can you guys hear?
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification.
So I do want to point out, I'll do a little bit of stopping, but basically I do want to point out that there is a 3' and a 5' adapter, which is going to be important later.
Additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a
So I just want to interject here that each of these columns, the lawn that they talk about, there's a physical number of places where stuff can bind. And so one of the limitations or, you know, a parameter that you need to consider is how much material do you want to start with in terms of catching on those little pieces, because the next step involves basically clonally like amplifying this sequence that you've captured. So you can either capture more original sequence and have less of the clonal pairs that then get sequenced, or you can start off with less and have more copies of it. And then that leads to the read depth that you get. So if you have like a million places stuff combined, you can essentially this comes down to how much multiplexing you can do. So you can put a bunch of samples and get like a shallower depth of read, or you can put less samples and get a deeper read depth. And so that'll become clear as you watch the next parts.
Of the hybridized fragment. The double-stranded molecule is denatured and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands.
I think this particular step is maybe new for this particular video is talking about paired end reading. So in this case, they're going to read the forward direction and then they're also going to read the other direction. And this can basically reduce the amount of errors, but because it's paired in reading, and again, you only have so many sites, you're going to reduce the read depth by half.
Reprime ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the...
And my understanding is this process is exactly the same as Sanger. In the case of Sanger sequencing, that nucleotide that gets put there blocks any further transcription, but in this case, they actually go in with enzymes and cleave off those things so that then another nucleotide can bind.
The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' end.
So the index is a way to tell you what RNA it came from, the original RNA, what sequence it came from. So this is how you can multiplex, is that each of them are going to have a unique identifier.
Of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse.
So then here you wouldn't necessarily have, you're gonna definitely have all the same, you should have pairs of forward and reverse reads if you did paired end reading, and these may not be symmetric if you are not doing paired end
Are
reading.
paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
So we've talked a little bit about reference genome. So obviously the better your reference genome is, the better this mapping is going to be. And in particular, if you have like a particular strain of mice and you're looking for SNPs, it becomes really important. And then one thing they don't show you here is that there are some reads that for whatever reason, maybe they're too short or something like that, they don't actually uniquely map. So you have to throw them out. So just FYI for this
The
part.
paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next generation sequence.
So I'm not like promoting Illumina or anything, but it was just one of a good, you know, basically a good video that showed how that works. So, so that's basically next gen sequencing, and it went through kind of rapidly sort of what the data analysis pipeline is, and I'm going to talk a little. So data analysis is not my strong suit. I have not actually personally done any of the data analysis, and so I'm not as much of an expert on that part. So that was basically you take the reads and you map them back, and you use the sequences to let you know if they should be aligning to the same one or not. And then there are some that aren't going to read, and then basically within a stretch of DNA. So in the case of shallow sequencing, you're going to maybe try to have coverage of more of the genome. And in the case of deep sequencing, you might actually get information about like how much of a gene is expressed. So in the case of people who are looking for SNPs, they're probably going to want really deep reads so they can actually say, was there less of this gene and more of this gene, to be a little bit more quantitative. And then essentially the unit that's used is reads per km, so kilomorgans, I forget what km is. But basically, it's taking sections of the DNA and saying how many reads did you get that mapped and tiled. So how deep was that stack that you assembled? So there are a lot of computational tools that are involved. So you can probably find those on, I didn't actually put a slide for the repositories that those are in, but I'll talk a little bit more about it with reference to single-cell-seq. But basically every workflow, because of the way that they work and the paired in readings and how the indices work and the clustering, they're going to have unique analysis pipelines as well. So you really have to pay attention to that when you're doing your data analysis. And then so we are going to talk a little bit about single-cell-seq, but there's another kind of trick that you can use to get cell type specific RNA and then use it for downstream sequencing that we just talked about, using immunoprecipitation of translating ribosomes. So one of those techniques is called TRAP. This is the other TRAP, or maybe it's, I think it's actually the first TRAP. TRAP version 1. Translating ribosome affinity purification, as well as ribotag. And then there's another way that you can immunoprecipitate translating ribosomes using phosphorylated S6 ribosomal subunit. So I'm not going to go into too much depth because we've already talked about the seq side of it, but basically just to introduce the idea of immunoprecipitation. Again, this is based on basically you have some antibodies conjugated to beads. Usually they're magnetic and you can just literally pull them down out of a sample. So for original TRAP, basically they had expressed one of the ribosomal subunits, 10A, L10A, and that was conjugated to EGFP. And so EGFP was here, and then you could basically isolate polyribosomes, which is different from isolating total RNA. You have to be really careful to get your ribosomes without disrupting them. So you pull down polyribosomes, they'll have some of these proteins. The proteins that got labeled will bind to your antibody-coated bead. So you have a bead, magnetic bead that's been conjugated to GFP, anti-GFP. It will pull down the ones that express GFP and not the others. And then basically you reverse this binding so you can get just your sorry, it's not total RNA, it's going to be poly-A RNA. So it's a little bit biased in terms of what you're going to actually pull down. It's only going to be what's associated with ribosomes. So just to clarify the difference there. And then in the case of ribotag, it's very similar, but in this case, instead of GFP, they're using an HA tag. They've chosen a different ribosomal subunit, L22. And in this case, it's a, they made a mouse. They described the mouse in this paper. So essentially, they have a Cree driver that's going to allow expression of the HA tagged L22 ribosomal subunit. And again, the principle is the same. You conjugate beads to anti-HA. You pull down the ribosomes that are from the cells that recombined, and the non-recombined cells express wild-type ribosomes that don't get pulled down. So here's a pro tip. I did actually, one of the people that interviewed for a job in the neuroscience department last year trapped cells. So you can use trapped cells to label your cells, and then you can trap them using immunoprecipitation. So just another, you know, whatever, trap squared method. And then I would definitely recommend, if you are doing any kind of ribosomal pull down, because essentially the steps are going to be the same involved in pulling down polyribosomes. I got the original protocol from Miriam before she published this Nature Protocols, but her original protocol was very well notated is not the word, annotated, I guess would be the word, but would definitely recommend, if you're actually going to do this, to read this protocols paper, because it's very in-depth, and it tells you exactly why you're doing all of the steps that you do and what would happen if you did not follow the steps. And it gives you pause points and all of the things that you want in a good molecular biology protocol. So then I don't know if you guys were around, Zachary Knight came by, I think maybe last year. So he was in the same lab, Nat Heintz's lab with Miriam, and a few years later published this paper that's similar, but instead of using a tagged ribosomal subunit, you can actually use some native components of the cell. So basically, if the cell is activated, it's going to lead to signaling pathways that allow phosphorylation at this S6 subunit of the ribosome. And this is obviously a polyphosphorylation, so I'm going to talk a little bit about why that's important. So it's not just one phosphorylation event you're trying to detect, it's many. So basically you can have a stimulus, you get cells where S6 is getting phosphorylated. And so very rapidly after behavior, if you lyse the cells and use an a specific anti-PS6 antibody conjugated to beads, now you can see your activated and inhibited markers. I have done a few of these experiments, and we definitely see activation and can validate what the markers should be. I'm not as convinced that you get de-enrichment in inactivated neurons. So it would be really great if someone could really improve on, that's one thing that we always miss. So one thing is all of these activity markers, because they're based on IEG expression, I forgot to make a slide about this, but they're all dependent on what cells actually show certain IEGs. So for example, if you just take a brain, an animal has done a behavior, and you look for ARC and FOS and June, different cells are going to express different ones. And that's based on either the type of activity that's coming into the cell, or if the cell is tonically active, it may not show such a big peak of CFOS, so it depends on the baseline activity of the neurons. And then it could just be which of those activity markers are actually expressed in the cell. So for example, I think one of the problems with RAM, because it's based on like piecing together different promoter areas, that it's biased for certain cells because they express certain markers. So we're always biasing toward a certain type of activity in a certain type of cell with each of the different methods. And then one thing that we're completely missing is inhibition, more or less, as far as I know. So that's what I have to say about that. And then he followed up on this with a paper where he showed neurons that were sensitive to heat in the medial preoptic area. So he got all these cells with PS6 at 37 degrees in this particular area, and he sequenced and found several markers. But essentially, in the first generation of the pull down, I think they were using some sort of like multiple antibody against multiple phosphorylation sites on the S6 tail. And then basically, I think that either a new antibody came out or just a different one that you didn't have to use certain blocking peptides for. So for this techniques, it's because you're looking at a native marker and you don't have an introduced marker, you really have to be careful about what antibody you're using. So if you actually use this, be careful what you are using. So that brings us to single cell RNA-seq. We may not get through all of this, and I also didn't go into too much depth, so if there are particular questions you have after we sort of introduce this quickly, let me know, because again, this is new. I haven't used it personally, but I can always do some more research or follow up for you if you have particular questions. So as far as single cell RNA-seq goes, you know, downstream, the RNA-seq part, pretty standard. So it really comes down to how are you getting these minute amounts of DNA isolated, and how do you keep them straight once you put them on the flow cell, and what kind of biases you can introduce into the system because of how you're doing it. So there are two primary ways, and again, this is really focused towards what are people doing in neurons. Single cell sequencing and then single nucleus sequencing. And we'll talk about why you might want to use one or the other. So basically, for either one of these approaches, you're going to have a step where you dissociate single cells. Obviously, dissociating cells that are in culture or something, or like are very homogeneous, like a liver, is going to be very different from trying to dissociate mature neurons that have processes that go all over the brain, dendrites, axons. You know, some of them are interneurons, some of them are not, some of them projecting. So dissociating cells is very tricky in adult brain. So there's some specifics you need to pay attention to. You're going to have for sure some kind of cell death. So there are some different ways that you can isolate cells, and we'll talk about that in the next slide, but a lot of times it's good, even if you're not using FACS, fluorescence assisted cell sorting, even if it's not the way that you're dissociating cells, it's good to potentially do a FACS sort based on a live and dead cell markers so that you're not sequencing dead cells, because that will introduce bias into the system. And then the next parts, after you get your cells isolated, there's some really specific things that have to happen in terms of tagging the RNA species, basically the transcriptome that comes out of a single cell. But then after that, the subsequent steps are fairly similar, but then the analysis part gets to be a gigantic pain in the you-know-what. So a lot of people are basically using, at this point, 10x genomics. So basically it's a machine, it does the fluidics for you, it separates the cells, it introduces the lysis buffer, blah, blah, blah. And then it basically is what it does in terms of tagging putting all the things you need on the individual cells, and then basically then you multiplex it and put it on the column that we already talked about. So what we already showed was the Illumina work through. And then comes the hard part where you get these amazing plots. So how do you capture those cells? I'm only going to do like 2 more slides because I know we're getting late on time, but I just wanted to introduce how you capture single cells and nuclei. So again, we already talked about fact sorting. In this case, you've got cells, some are labeled or unlabeled, and usually you can do at least 2 markers. And so you can either have cells that are tagged and not tagged and sequence both, or you can have a marker for live cells and dead cells, and you can sequence the live cells and put the dead cells in the trash. So then another way that you can dissociate cells would be laser capture microdissection. So in this case, you're getting thin sections, and then you just use a laser to cut out a single cell. So this may or may not lead to a single cell generation, but there are computational ways essentially to determine if you've got a singlet or a doublet. But we can talk about that later. The one that really made headlines was the microfluidics paradigm, where they basically have the beads with all of the barcodes, and those are being flowed in to single cells, and then you basically get a droplet that contains a single cell along with the lysis buffer and the probes that bind the unique molecular identifiers onto the cells. And so I think this requires a fair amount of troubleshooting, because it really comes down to like the flow rate, and if the cell type that you've put in suspension flows, and then I think this is more susceptible to doublet formation than probably some of the others. And then if you have cells, for example, on a monolayer, or you have them in suspension after you've done the first dissociation of cells, you can literally go in and just pick up single cells. So that's a super manual way to get it. And then as far as getting single nuclei, you don't need a live nucleus. The nucleus is sort of its inside its own membrane. So you basically lyse the cells, pellet down the nuclear fraction, and then you can fact sort the nuclei. So obviously with the nuclei, you're not going to get all RNAs. You're going to lose any that were in the cytosol, but there have been some studies that show that basically the nucleus is representative of what's in the cytosol being translated.
So Anita, question. So why would you choose to do full cells versus nuclei?
So essentially, I think my recollection is that depending, so in Catherine's lab, there were people who were trying to improve the dissociation method. And so they improved it such that they were getting more cell survival. The conditions under which more cells survived were biased towards certain cell types. So the endpoint data and then the clustering showed that like certain clusters were very small. So for example, I think one of them that was small it wasn't oligodendrocytes, because I think they actually had quite a lot of those, but maybe something like microglia. And then you have a whole cell category because they're susceptible to the dissociation. And then you filter them out. If they die, you're losing that piece of your data. So in the case of the nucleus, you're not worried about losing stuff because it's dead. You can sequence everything. But it's going to bias it in a different way. I was like, where did my slide go? I have some weird animation on here. So here's just I'm not going to explain. We'll go into like drops. I have drop-seq and then some applications of drop-seq, and then I can go into more detail next time about the workflow, but this is the behemoth workflow that is necessary for your single cell. And then some of you guys are taking the Python class, and then we'll talk about in situ hybridization. So we can finish this up next time, and then I'll also talk about high throughput microscopy and brain clearing for our next class. So I apologize for going over, but I wanted to give you guys kind of a broad range of technologies. And probably I've been talking too much for you to ask questions, but if anybody has questions or there are things you want me to go into more detail on, just email and I will address those for next time. Or if anybody has questions the last couple minutes. All right, you guys know who you're going to pair up with for your presentations? Okay. All right.
How many more do we have left? Classes? Is it like 2 more?
1 more lecture, and then next Tuesday is the presentations.
Okay.
All right. Thanks, everyone. Have a good one.
Thank you.
Thank you.
Thank you.
Thank you.